Hello friends, I am BP Shah, PGT Bio, Kendriya Vidyalaya Keshapuram. Today I am going to continue my last session lecture, that is respiration in plant, chapter 14 of plant physiology, unit 5 of class 11th NCERT. In my last session, I have discussed about various types of respiratory substrate and their RQ, and I have given some questions. Today I hope now you all are able to answer these. These questions which I have already previously asked are: what are the differences between respiration and combustion? Because in both the process energy is liberated. The answer is respiration occurs in living cell only, but combustion in non-cellular process. Second point is it is under biological control, mainly it is controlled by enzymes. Combustion is an uncontrolled process. Third point is respiration is a biochemical process, while combustion is physiological process. Fourth point is a number of enzymes is required, one for each step of the reaction. There are various type of reaction takes place in the respiration like glycolysis, Krebs cycle, electron transport chain, oxidative phosphorylation. All these reaction requires various type of enzyme in each steps. In case of combustion, there is no use of enzyme, so it is non-enzymatic process. In respiration, next point is ATP is generated. ATP, the full form is adenosine triphosphate. It is made up of by adenine, ribose sugar and 3 phosphate group, which is known as energy currency, which gives energy when the cell needed this. But in case of combustion, heat energy liberated, ATP is not generated. Enzyme is required, as we have already discussed. In case of combustion, there is no enzyme. In my second questions, what are the respiratory substrate? Name the most common respiratory substrate. Respiratory substrate are those substances which help in generate ATP. There are various types of respiratory substrate molecule by which cellular organism release their energy. The answer is respiratory substrate are the organic substance which are oxidized during the respiration, which liberate energy inside the living cell. The second question is the most common respiratory substrates are carbohydrate, especially glucose, which is the product of photosynthesis, which I have already discussed in my last lecture, acts as a respiratory substrate. Besides this, protein, fat, organic acids also acts as a respiratory substrate. Now, my third question is, what do you mean by RQ and what is its value for fat? RQ, as we have already discussed, it is respiratory quotient and it is defined as volume of CO2 evolved divided by volume of O2 consumed. As we have already known, the different type of respiratory substrate, RQ value is different, it depend upon the what type of respiratory substrate is used. In case of glucose molecule, the RQ is 1, for fat and proteins RQ is less than 1. In fat and protein, for their oxidation it require more amount of oxygen. Here the reaction is the tripalmitic acid, that is the fatty acid, it required for the oxidation 145 oxygen. So, for the oxidation of fat as well as protein, it require much amount of oxygen in comparison to the carbohydrate like glucose. So, the RQ of fat is less than 1 in comparison to the carbohydrate. So, this is the last session questions. Today I am going to continue my chapter, that is respiration in plant, and the major steps are glycolysis, which I have already discussed later on. Here is the questions, because the chapter name is respiration in plant. Why plant do not have a special type of respiratory organ? Because in your class 6 and 7 you have already known that there are various types of respiratory organ in cockroach, that is tracheal systems are there. In case of frog, the moist skins are there. So, various type of specialized respiratory organ in case of various types of organism, but in case of plant it is not needed, not a special type of respiratory organs are there, because each part of the plant can take care of its gas exchange demand. Like leaf, there is the presence of stomata. Stomata help in the exchange of carbon dioxide and water and oxygen. The second point is they do not have much demand of gaseous exchange, because for the respiration it require oxygen. This oxygen need fulfilled by the photosynthesis product. In that cases the carbohydrate molecule is formed and oxygen acts as a byproduct. Carbon dioxide combined with the water and formation of carbohydrate, water and oxygen. So, to fulfill the oxygen demand for respiration, it is already present in the plant with the help of photosynthesis. Plant produce oxygen by the photosynthesis. Next is root, stem and leaf respire at low rate in case of the animals. Each living cell in the plant located close to the surface of the plant. So, plant do not have a special type of respiratory organ in comparison to the other animal cell. Now, respiration is a amphibolic process. Amphibolic means where the catabolic and anabolic process takes place. Generally, the reaction of the respiration just depicted that here the glucose molecule with the help of oxygen, if the oxygen present, it converted into carbon dioxide, water and ATP. If the oxygen present, then carbon dioxide and water is formed and ATP is formed. To see this reaction, it shows that this is the catabolic reaction. Because here the large molecules break down to the smaller one, but In the stepwise reaction of the glycolysis and various other part like Krebs cycle and electron transport chain, various other materials is formed which help in the formation of fat and protein like alpha ketogluteric acid, one of the product in case of Krebs cycle, it help in the formation of fatty acid and proteins. Protein is also formed. Succinic acid and malic acid is the product which helps in the formation of protein. So, it also help in the synthesis as well as in the breakdown. So, this one is called amphibolic process. Here the respiratory pathway is both catabolic and anabolic, we have already discussed. Various substrates are broken down driving their energy. During the synthesis of fatty acid, acetyl coenzyme is withdrawn from the respiratory pathway. Krebs cycle, which is the common metabolic pathway in the interconversion of carbohydrate, protein and fat. If the carbohydrate is not present, protein and fat also helps in the respiratory pathway. This depict the how the fat, carbohydrate and protein help in the formation of other reactions in carboxylation process like oxidative carboxylation, where the pyruvic acid converted into the acetyl coenzyme. Here is the fat is converted into fatty acid and glycerol, then fatty acid directly form the acetyl coenzyme, which is the link product, because the pyruvic acid are formed inside the cytoplasm and the pyruvic acid directly not enter into the mitochondria. Firstly, it converted into 2 carbon compound, that is acetyl coenzyme, then it allowed to enter into the mitochondria for the Krebs cycle. Carbohydrate help in the entrance of pyruvic acid and acetyl coenzyme to the Krebs cycle, and protein converted into amino acid, then it converted into pyruvic acid, then acetyl coenzyme. So, fat, carbohydrate and protein that also help in the respiratory pathway instead of glucose molecule. There are various types of process in the respiration, that is glycolysis, Krebs cycle, electron transport chain and oxidative phosphorylation. The glycolysis, as we have this is the first steps of the respiration. This glycolysis help in the conversion of glucose molecule into pyruvic acid. The glycolysis is the process by which 6 carbon compound, that is glucose, it is a type of carbohydrate, is converted into 2 molecules of 3 carbon compound, that is pyruvic acid. The term glycolysis, it is originated from the Greek word glykos, which means sugar. We know that in my last class the product of photosynthesis is sugar, which is a disaccharide and formed by glucose and fructose. The first term glycose means sugar and second term lysis, which means breakdown or splitting. As the term indicate, glycolysis is the process in which sugar molecule which is already present in plant, the end product of the photosynthesis, and converted into 3 carbon compound molecule of pyruvic acid. It is a series of reactions that was given by 3 scientists, Gustav, Imbaden, Otto Meyerhoff and J. Parnas. As the name of the 3 scientists, the first name from the Imbaden, M from the Meyerhoff, P from the Parnas. So, the glycolytic pathway also known as EMP pathway. This is the only process in case of anaerobic respiratory organism. Finally, we will see that it liberates energy in the form of ATP and 2 ATP is formed. So, these 3 scientists gave the various type of systematic process in which glycolysis takes place. In the sequential reaction of the glycolysis, firstly the sucrose which is the product of photosynthesis, with the help of enzyme invertase is converted into glucose plus fructose. Both are 6 carbon compound, it is the carbon 12 compound. It converted into C6 and C6. Glucose is the aldehyde molecule, fructose is the ketone molecule. Both glucose and fructose it enter into the glycolytic pathway. The first step of glycolytic pathway, that is both glucose and fructose molecule, it is going to be phosphorylated. Phosphorylation takes place. There are 3 types of phosphorylation which we discussed in my subsequent lecture. Oxidative phosphorylation, photophosphorylation we have already discussed, formation of ATP with the help of light energy, and the third one substrate level phosphorylation. Where the substrate molecules form the ATP. This substrate level phosphorylation we will see in Krebs cycle. So, first of all glucose molecule and fructose molecule utilize ATP and convert it into glucose 6 phosphate. Glycolysis, as we have already told, it is a series of reaction and controlled by different types of enzymes, and there is utilization of ATP as well as NADH for the source of hydrogen. Firstly, it is well to know that what is the composition of ATP, as the term, because in various steps of the respiration ATP either consumed or it is formed. So, firstly discuss about the ATP. The full form of ATP that is adenosine triphosphate. Adenosine, which is made by nitrogenous base, that is adenine, which is a purine, two ring structure, this type of structure is there, one ring and the another ring, two ring structure, and it combined with the sugar, pentose sugar, that is ribose, or it may be deoxyribose, and it further that form adenosine, adenine plus ribose sugar. Then the 3 phosphate molecules combined with this. The phosphate molecule are present in the form of phosphoric acid H3PO4, that is phosphoric acid. Simply we can say the ATP is formed by adenosine, adenine, sugar and 3 phosphate molecules. Which are in the form of ATP. The liberation of the last phosphate molecule, it release huge amount of energy, then it form adenine, sugar, 2 phosphate molecule, only 2 phosphate molecules are there, then it liberate huge amount of energy. This energy are used in various metabolic process. And for after the liberation of last phosphate group, it form ADP, adenosine diphosphate. If the second phosphate molecule is removed, it also release energy, but less in comparison to the first. Then now the structure will be adenine, sugar, and phosphate, it release energy. Now, it is called AMP, because one phosphate is there, that is called monophosphate, 2 phosphate is there, diphosphate, 3 phosphate is there, that is called ATP. So, this is the basic concept of ATP, ADP and AMP. We can relate all three. Now, the structure of the ATP molecules are as we have already discussed. The adenine is a type of purine which is 2 ring structure, that one ring, this one ring is called pyrimidine ring, and here and the another ring is also attached with the first one. This one is called imidazole ring. The adenine, which is symbolized by 1, here its number will be 1, 2, 3, 4, 5 and 6, this one 7, this one 8, this one 9. So, adenine molecule have 9 atom in this sequence, and at 6 carbon there is the amino group. So, we can say adenine chemically it is 6 amino purine. And from this 9 atom, the ribose sugar is attached. Ribose sugar, which is 5 carbon compound, and this ribose sugar is attached, and the number will be this type 1, 2, 3, 4, here is the 5. The first carbon of ribose attached with the 9th atom of nitrogen of the adenine, then 3 phosphate group is going to be attached in the form of phosphoric acid. H3PO4 is written in this form. So, you can easily understand. So, this is the reaction of, this is the structure of ATP, which have 3 phosphate molecule. This phosphate molecule, as we have discussed, it liberates energy when the cell is needed. In the entire process of the glycolysis, we will find that there is 2 ATP is used and 4 ATP is formed. Glycolysis, it takes place in cytoplasm. Generally, in the all aerobic or anaerobic organism, but in case of yeast and RBC, red blood cell, it occurs in plasma lemma. It is the process in which takes place without oxygen. In entire step of glycolysis, we will find that there is no use of oxygen. And the second thing is, no carbon dioxide is produced. So, there is no decarboxylation. No decarboxylation process takes place in the glycolysis. It takes place mainly in the cytoplasm, and all the anaerobic organism have ATP in this form. Now, there are the various types of steps in the glycolysis. Mainly there are 10 steps. Now, first of discussed steps involved in glycolysis. The steps are, first step is called phosphorylation. As we have discussed, phosphorylation means formation of ATP or consumption of ATP. The second process, the second step in the glycolysis is isomerization, where the glucose molecule is converted into fructose 1 with the help of enzyme isomerase. The third step is again phosphorylation, and fourth step is breakdown, that is cleavage. Here the fructose 1,6 diphosphate we will see breakdown into the 2 molecules of 3 carbon compound. The fifth step again phosphorylation, and sixth step is dephosphorylation, and seventh step is rearrangement. Here no molecule is formed or taken in, but only the rearrangement takes place. The eighth one step is dehydration. As the term indicate, here the liberation of water molecule, and ninth and last step is dephosphorylation. These entire steps it require 2 ATP and produced 4 ATP. So, 2 ATP is lost and 4 ATP is formed. So, the total gain is 4 minus 2, 2 ATP is formed. Besides that, there is the reduced coenzyme, that is NADP, 2 molecule of NADH is formed, which is going to be further reoxidized in oxidative phosphorylation, and for 1 NADH 2 it gives 3 ATP. So, after the reoxidation of 2 molecule of NADH, it forms 6 ATP, which will be find later on. Now, what are the different types of steps when we see in the form of biochemical reactions? So, we know that the glucose molecule is a aldehydic molecule and made by 6 carbon. So, for the examination of point of view, it is easily to write 6 carbon firstly, 1, 2, 3, 4, 5, 6, and here the arrangement of H and OH, H and OH. At the 6th carbon there is CH2OH. So, this is the glucose, a 6 carbon compound, aldehydic form are there. Now, firstly we have already discussed glucose first of all converted into glucose 6 phosphate. It is easy to write the formula of glucose 6 phosphate at 6 position. Here the number of carbon we can 1, 2, 3, 4, 5, 6. At the 6th carbon instead of H there is phosphate group. So, it is easy to write CHO HOH as second, H at third, in fourth HOH, in fifth HOH, in sixth there is change CH2OP. We can write the phosphate group are there. So, this is the structure of glucose 6 phosphate. For this reaction, the cell required enzyme, that is hexokinase, and Mg plus 2 plus plus. In this reaction, the ATP which already discussed acts as a energy currency. Here energy is needed. The ATP adenosine triphosphate it converted into ADP. So, in first step 1 ATP is consumed. So, we can write in the form of ATP consumption the glucose 6 phosphate converted into, when we find the glucose account, finally, it is easy to learn ATP account during the glycolysis. Here the serial number, stage, ATP released, ATP consumed and net gain. We have seen when the glucose molecule converted into glucose 6 phosphate, 1 ATP is consumed. So, there is the loss of 1 ATP. We further see the when the ATP molecule is used or liberate. The second step, the glucose 6 phosphate is converted into fructose 6 phosphate. as we know glucose and fructose have same formula, but the glucose molecule is aldehyde and the fructose one is ketone. So, it will write as here the ketonic group CO fifth same HOH and sixth one will be the same. The conversion of aldehyde into the ketonic form. So, the structure will change glucose into fructose 6 phosphate. This is the reaction called isomerase. Isomerization takes place, the enzyme will involve isomerase. Here the substrate is glucose 6 phosphate. So, we can say the enzyme is phosphohexoisomerase. With the help of this enzyme, the glucose 6 phosphate molecule is converted into fructose 6 phosphate molecule. Now, the fructose 6 phosphate molecule, it is converted into fructose 1,6 bisphosphate. As the one phosphate molecule present at the 1 carbon and another one at 6 carbon has already present. So, it require ATP. Here one ATP is also consumed and ATP converted into ADP. Here the enzyme is fructokinase. We can say it as phosphofructokinase, because phosphate group already present as 6 carbon atom of the fructose molecule. So, that we can easily write it as fructose formula is there CH2O. We have already discussed at the 1 carbon phosphorus is there, and next is same as fructose 6 phosphate. Fifth is same and sixth one here, sorry, here also oxygen is there, because in fructose there is CH2OH instead of H, phosphate is there. So, this is the fructose 1,6 bisphosphate. By adding and subtracting some molecule, we can easily write down the formula of every steps. Here the first and sixth have phosphate group. Now, I have already discussed the next step is cleavage. Cleavage means breakdown. Here the 6 carbon molecules is going to be break down into 2 molecules of 3 carbon and 3 carbon, that is glyceraldehyde 3 phosphate 3 phosphate and dihydroxy acetone phosphate. This glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, it is a type of carbohydrate that is called triose. The glyceraldehyde 3 phosphate is aldehydic and dihydroxyacetone phosphate are found in the ketonic form. So, it is converted into aldehyde. We can easily write 3 carbon CH the first carbon have aldehyde group, the second one HOH and the third one CH2OP. That is, at 3 carbon there is a phosphate group. The dihydroxyacetone phosphate also easily write CH2OH, C double bond O and CH2OP. Here at 3 carbon phosphate group is in glyceraldehyde and 3 carbon phosphate group are present in the dihydroxyacetone phosphate. Now, this glyceraldehyde 3 phosphate and dihydroxyacetone phosphate are easily interconvertible. It can easily convert it into one form to the another form. So, after that this glyceraldehyde molecule, it going to be further reaction and for 1,3 by phosphoglycerate. Because the dihydroxyacetone phosphate also converted into glyceraldehyde. So, this 1,3 biphosphate glycerate, it is formed in the 2 molecules. 2 molecules of 1,3 biphosphate glycerate molecule is formed. It required enzyme that is called dehydrogenase. And 1 phosphate molecules are also required in the form of H3PO4 and NAD is also required, which converted into NADH2. Now, the 1,3 diphospho या biphospho, the older name is di. The biphosphate is structure is COOP at second carbon CHOH, at third one CH2OP. So, this is the structure of 1,3 biphosphate glycerate. This 1,3 biphosphate glycerate, it gives two molecules of 3 PGA, that is phosphoglyceric acid. This PGA 3 phosphoglyceric acid, we can write it COOH, CHOH, CH2OP. In 3 phosphoglyceric acid at carbon third phosphate group is there, one phosphate which is found in the first carbon at 1,3 bisphosphate now converted into COOH. It release, the phosphate molecule is released, this phosphate is combined with ADP, adenosine diphosphate, which we already discussed. Now, it converted into ATP. So, here is the 2 molecules are there. So, 2 ATP is formed. Now, the enzyme, as we have already know where the ATP is used, enzyme is involved, that is kinase. Here the name is phosphoglycerokinase, and this one NAD, it combined with hydrogen, it form NADH2. NADH2. NAD is a coenzyme, it is the acceptor of hydrogen atom, and it form, after the combination of hydrogen atom, it form NADH2, which further reaction in the oxidative phosphorylation or in electron transport chain, it liberate energy. So, we can summarize it in the form of this, the ATP account. The first we have already seen, the glucose molecules converted into glucose 6 phosphate, it consumed 1 ATP. So, there is the loss of 1 ATP. In the second step, fructose 6 phosphate converted into fructose 1,6 bis phosphate, it also required 1 ATP, that is another loss of 1 ATP. So, finally, there is the loss of 2 ATP in this 2 stage. We have already discussed at the time of conversion of 3 phosphoglyceraldehyde, when it converted into 1,3 by PGA, 2 molecules of NADH2 is released. As we have seen later on, this NADH2 it released, one NADH2 released 3 ATP. So, there is the formation of 3 into 2, 6 ATP will be formed in the latter stage of the oxidative phosphorylation. And the second step, next stage is 1,3 by PGA is converted into 3 PGA, it released 1 ATP. 1 ATP in the form of 2 molecules, it gives 1 into 2, that is there is a gain of 2 ATP. So, these are the 4 steps where ATP consumed, ATP released, and the further reaction is this 3 PGA. Now, this 3 PGA now converted into 3 phosphoglycic molecule, it now converted into 2 PGA. This it requires enzyme that is called mutase, and this structure is COOH, CH2OH. The 2 PGA same structure as the 3 PGA, here the at 3 carbon phosphate is there, now it is shifted to the second carbon. This 2 PGA now converted into PEP, phosphoenol pyruvate. In this reaction, water molecule is released. So, the process is dehydration, process takes place, and PEP finally converted into pyruvic acid, which is the end product of the glycolysis. So, at the time of when PEP converted into pyruvic acid, it also release phosphate molecule, that is converted ADP into ATP. So, 2 ATP is also formed here. So, the final step of the ATP account, 2 phosphoenol pyruvate, it converted into pyruvic acid, it released 1 ATP, that means 2 molecule of pyruvic acid is formed. So, there is the 2 ATP. So, final form is 2 ATP plus 2 ATP, 4 ATP is gained, 2 ATP is lost. So, net gain of direct ATP is 2 ATP. So, we can say that in glycolysis 2 ATP is formed and further reaction 6 ATP we will get at the later stage of the oxidative phosphorylation. So, in this process the entire process of the glycolysis which takes place in the cytoplasm, the ATP account which have already seen, and the final product is pyruvic acid which further on goes to the further reaction that is called Krebs cycle and electron transport chain, oxidative phosphorylation, so on. In case of anaerobic organism, these 2 steps, these 2 ATP is formed and further gone into oxidative decarboxylation. So, this is the entire thing of the glycolysis. Now, you can test yourself what are the main steps of aerobic respiration, what is the net ATP production in the glycolysis, in which cell organelles does 2 times does is repeated, respiration takes place, and where does aerobic and anaerobic respiration takes place in a cell. Now, I hope you are able to answer these questions. It will discussed on the later stage. Thank you.